Genomic sequencing is the technology that allows us to know the information on the DNA of a particular sample, in this case a virus. Genomic surveillance is how we translate that data with epidemiological data into knowledge. My name is Sonia Gonçalves and I am responsible for the delivery of the Genomic Surveillance Service for COVID operations at the Wellcome Sanger Institute. Genomic surveillance is important because it helps us understand what is happening in the population of a virus. Without genomic surveillance, we are going a bit blind because we don't know what is happening and what is, for example, the cause of an increase of cases or a decrease of cases. From the beginning of the pandemic, we were looking at specific use cases. For example, how was the virus being introduced in the country, how the virus was spreading within the UK, and how could we control outbreaks by using the genomic surveillance data. More recently, we are more focused on the identification of the variants of concern. A variant of concern is when a mutation or a group of mutation is associated with a difference on how the virus behaves. For example, if that specific variant has increased transmissibility, or if that specific variant has an impact on the immune response. Here at Sanger, we receive between 400,000 and 500,000 positive and negative samples in a week, and we sequence at the moment around 20,000 samples per week. From the genomic surveillance point of view, as soon as a variant of concern is detected, this information is then provided to the public health agencies that use that information to contain the spread of those variants. So the genomic data that is generated through the genomic surveillance operations is immediately shared at a national level, at international level. The sharing of the data in a pandemic is absolutely fundamental on how we control the pandemic, because a variant of concern appearing in one part of the world affects the whole world. So the activities of genomic surveillance with COVID have been an incredible learning experience. We are still learning as we go, and we will still be learning even after the pandemic ends and looking back and learning for all the data that was generated during the pandemic. But I think we have built the resources, we have built the infrastructure, we have built the analytical methods, so we will be better prepared to respond to future pandemics.
When the COVID-19 pandemic began, researchers and public health experts warned us that the earliest possible window for a vaccine would be the end of 2020. They also cautioned us that vaccine development takes time, and it could be much longer than that. But in the closing weeks of the year, two vaccines, one from pharma companies Pfizer and BioNTech, one from Moderna, began rolling out in some parts of the world. They weren't the first worldwide, but they were, in a sense, the first of their kind. And like Babe Ruth calling his shot, it seems a little like the experts knew how this was gonna go. And that's because a technology decades in the making was finally able to rise to the occasion just when we needed it most. This is the story of how a new vaccine technology based on RNA came to be. And if it continues to prove safe and effective, it won't just be for COVID, it will be a major change in the way we design all vaccines in the future. Now we're gonna cover a lot of research today, but none of these papers were published out of the blue. A lot of progress in immunology and biotech had to happen for mRNA vaccines to happen, and a lot of people had to do that research. The job of a vaccine is to safely expose our immune system to an antigen, a piece of a protein from a pathogen or infectious agent that our immune system will remember and recognize. It's like a wanted poster that will teach our immune cells what to seek out and destroy when a real infection happens. Traditionally, we've introduced antigens in a few different ways, using a live weakened pathogen, so one that is alive but won't hurt us, a killed pathogen, or just a piece of one. We've also used viruses to deliver instructions to our cells to make an antigen. Whatever method you use, this takes years of work. For example, to make the measles vaccine, scientists had to grow the virus for almost 10 years. They needed to weaken the virus enough that it would trigger an immune response without making you sick. But starting around the 1990s, scientists thought that maybe they could cut out the middleman and use messenger RNA, or mRNA, to reprogram our cells so that they make those viral antigens by themselves. Instead of us producing them in a laboratory somewhere, our cells could do the work. They hoped that such an approach might be safer and more efficient than traditional vaccines, at least for some diseases. After all, the job of RNA is to guide the production of proteins in a cell, and antigens are generally proteins. But mRNA isn't actually the genetic material inside our cells. That's DNA. You can think of DNA as a giant library containing the blueprints for any kind of protein your body might need to make. But since it doesn't make sense to schlep the whole library with you every time you need to ask a manufacturing plant to make something, it is easier just to copy out the specific protein blueprints you want. mRNA is that copy. It brings the genetic sequence for a protein transcribed from the cell's DNA to the place where proteins are made. mRNA vaccines use this feature to safely coax our cells into using their own protein-making machinery to create a viral antigen from scratch. And this turns out to be a big advantage when you're dealing with something like a totally new virus causing a sudden pandemic. Because designing one of these vaccines doesn't even require a sample of the virus. All you need is a digital file with its genetic sequence. That's because as long as you know the sequence of DNA or RNA, you could just make it. It is not nearly this simple with protein-based antigens. Proteins are all foldy and weird. DNA and RNA are just linear strings. Scientists can simply download the genetic sequence of the virus and have a candidate vaccine ready to start testing within weeks or even days. That's what happened with Moderna's vaccine, which was ready for preliminary tests less than a month after the genome of the SARS-CoV-2 virus was published online. Also, this enables a plug-and-play approach. Once you have all the basic pieces to make an mRNA vaccine in place, you don't need a new setup to make a new vaccine for a new virus. Theoretically, you could just swap in new RNA and go from there. And there's also one more reason they're so speedy to put together. Many vaccines require adjuvants. These are substances that enhance the immune system's response to the vaccine and attract the right immune cells. But mRNA, it turns out, is pretty good at bringing in the immune system all by itself. So it avoids the potential need to spend additional months or years testing various types and amounts of adjuvants and whether they're necessary to make the vaccine work. You can see how all of this could make mRNA vaccines the perfect technology to rely on when we need a defense against a new pandemic stat. But there's a reason why we're only now hearing about them, because they just weren't ready before. You see, for all its benefits, mRNA also has some drawbacks, which have taken literal decades of research to resolve, just in time for COVID-19. This research got its start around 1971 when UK-based researchers studying protein production put mRNA from a rabbit into frog egg cells. They found that those cells produced the rabbit version of that protein thanks to the mRNA code. This led to a series of similar experiments with scientists being able to insert mRNA into more and more complex types of cells. Researchers also kept working on efficient ways to deliver mRNA into a cell. The first experiments in using mRNA as an actual vaccine started taking place in the early 1990s, and that is where researchers ran into huge problems. The major roadblock was that when it's introduced into the body, RNA can be pretty hard to keep in one piece. It turns out free-floating RNA is often used by tumor cells to make it easier for them to spread around. RNA that hangs around outside of our cells can also be a remnant of a cell that was infected by a virus and then blasted apart by the immune system. And so to keep us healthy from those two things, our bodies have a lot of ribonucleases, which are enzymes that break up free-floating RNA to get rid of any potential danger. So that's why in early experiments, mRNA would get destroyed before enough of it could get into a cell and start doing its magic. This problem stymied mRNA vaccine research for decades, until scientists found ways to make mRNA more stable. One solution was adding specific gene sequences to cap the beginning and the end of the mRNA strand. That made it look more like mRNA that was generated by our own body. But that wasn't the only quirk of messenger RNA that scientists had to contend with. On top of the ribonuclease problem, free-floating RNA can activate the immune system and attract it to its location destruction. Yeah, sure, like we said before, attracting the immune system can be helpful because you need that to happen for a vaccine to work anyway. But this was too much of a good thing. In early attempts, the mRNA was activating the immune system so much that it would clear away the vaccine before it could do its job. Like, the goal of using an mRNA vaccine is to teach the immune system to seek out the antigens that the vaccine will program our cells to make, not to destroy the message before it gets a chance to do anything. And then we reached 2005 when researchers discovered the secret handshake that allows our body's RNA to avoid immune destruction. See, RNA is composed of 4 chemical bases which mirror those used in DNA. But it turns out that in mammals, a lot of those bases are chemically modified until the RNA strand is needed to guide the creation of a protein. This is not the case in most pathogens. That's why when the immune system notices a strand of unmodified RNA, it's a clear sign that it's dealing with an invader, and then it's time to mount an attack. Figuring this out meant that scientists could now apply those chemical modifications to manufactured RNA. In fact, it actually made mRNA vaccine technology more customizable. Basically, researchers could tweak the percentage of modified bases in the mRNA just enough to call the immune system to the area, but not enough to induce an all-out attack and deactivate the vaccine before it can start helping your body. All right, we have done a lot of work here. From the 1970s to the early 2000s, the final challenge that scientists had to overcome was how to deliver the vaccine into a cell. The mRNA molecule itself is too big to get through a cell's membrane easily. Experiments demonstrated that some can sneak in, but not enough that you could just throw it at cells and hope for the best. Now, there are specialized ways to introduce nucleic acids into cells in a lab setting, but they aren't always suitable for use in like a living human body. Things like zapping the cells with electricity to open little holes to let things in. It's not that these methods can't be adapted for use in humans, it's just that there are better options than zapping people. A simple injection is what we want, something people are used to, also something we have all of the technology already to administer, especially if you want to fairly quickly vaccinate billions of people. And that's why scientists eventually turned to lipid nanoparticles, which are the delivery method used in the first 2 mRNA vaccines to hit the market. Lipid nanoparticles, or LNPs, are tiny balls of layered lipids, or fats, with an mRNA payload tucked safely inside. The LNPs have a positive charge, which makes them stick to the negatively charged cell membranes. In a process called endocytosis, the cell then wraps the LNP in a piece of its membrane and swallows the package. Once inside, our cellular machinery unpacks the whole package, and the mRNA can start making the antigen proteins necessary to train our immune system. And this isn't that new of a technology. As early as 1978, scientists were able to use a basic version of these tiny balls of fat to get the mRNA inside mouse spleen cells and make them synthesize a new protein. Early LNPs had some issues with efficacy, but researchers eventually perfected the technology just a few years before it turned out to be needed to quickly develop an mRNA vaccine for the COVID-19 pandemic. Building on successful studies delivering other types of RNA into cells, in the early 2000s, researchers started experimenting with LNPs as ways to make mRNA vaccines easily injectable. And in 2018, the FDA approved the first RNA drug that also used LNPs, which means the delivery vehicle was ready just in time when researchers started working on mRNA vaccines for COVID-19. So even though the Pfizer-BioNTech and Moderna COVID-19 vaccines were the first mRNA vaccines to be authorized, researchers had been excited about the potential of this technology and had been working on it for decades. It's true that we've thrown a lot of money and person hours at stopping this pandemic, and without that investment, we probably would not be seeing mRNA vaccines just yet. Some efforts to create other mRNA vaccines had already been abandoned, and mRNA vaccines were never considered a sure thing. We had to do the testing, which is why it took until late 2020, but there was an existing body of research to draw from, including into other major coronavirus diseases, SARS and MERS. What researchers learned allowed them to create these new COVID vaccines even faster. In one sense, these COVID vaccines were an explosive development, an incredibly swift global collaboration in the name of human health, something that feels to me on the scale of an Apollo mission. But all science is incremental. It always builds on the dedicated work of generations of researchers, and it never happens in a vacuum. In fact, multiple mRNA vaccines for other viral and bacterial diseases, and even for some cancers, are undergoing human trials. Now that mRNA vaccines are working, it's likely they will keep working, and that is great news for all of us.
Vaccines are about to change the world again. For generations, vaccines have helped us live longer, safer lives, protecting us from smallpox, polio, measles, and other devastating diseases. Now, a new type of vaccine can make even more diseases, from HIV to coronaviruses, a thing of the past. Vaccines usually work like this. A tiny part of a virus, called an antigen, is injected into your body. It's not enough to make you sick, but it's just enough to make your immune system notice. Your immune system then creates antibodies, soldier cells that are trained to fight that particular antigen. Once this happens, your body will remember how to quickly produce more antibodies if needed. So if you're ever exposed to the real virus, your body will already know exactly how to defend itself. But what if you could skip that part and just train your immune system to make antibodies for specific diseases? That's what mRNA vaccines do. mRNA stands for messenger RNA. It's a piece of genetic code that tells our cells what to do, like make new proteins or repair damage. mRNA is naturally found in all of our cells. But in 2005, infectious disease experts at Penn Medicine discovered how to modify mRNA to safely make our cells create proteins similar to those found in some viruses. Once those proteins are made, our immune systems can then create antibodies for that virus. Today, this same technology is being used by scientists around the world to develop vaccines for all kinds of diseases, including coronaviruses that could cause future pandemics. Researchers at Penn Medicine are developing mRNA vaccines for diseases like herpes, HIV, sickle cell anemia, and even cancer. And as we continue to push for new breakthroughs in mRNA technology, we're paving the way for a safer, healthier future for all of us.
mRNA vaccines for COVID-19. Vaccines are substances that protect you from harmful diseases. Most vaccines contain parts of weakened or dead germs that trigger your immune system to fight the disease. But mRNA vaccines for COVID-19 are different. They contain a substance called mRNA that teaches your cells how to make a protein that triggers an immune response. In order to understand how these vaccines work, it's important to know what mRNA is and how it normally makes proteins your body needs. Most cells in your body have a command center inside them called the nucleus. It contains genetic material called DNA that consists of instructions for building and maintaining your body. Proteins are one of the building blocks of your body. When a new body protein needs to be built, instructions for building it are copied from your cell's DNA and converted into a message called messenger RNA, or mRNA. Then the mRNA travels out of the nucleus to a protein-building machine in your cell called a ribosome. As the ribosome reads the message from the mRNA, it builds the protein your body needs. mRNA vaccines take advantage of this process to help give you immunity to COVID-19. Each vaccine contains special mRNA that provides instructions for your cells to build a harmless piece of the virus called the spike protein. The spike protein is found on the surface of the SARS-CoV-2 coronavirus that causes COVID-19. Each piece of the mRNA from the vaccine is wrapped in a protective coating. The vaccine is given as a shot in the upper arm. In the body, the mRNA particles enter your cells. Once inside the cell, the mRNA travels to a ribosome. Using the mRNA from the vaccine, the ribosome makes only a piece of the spike protein from the virus. After making the piece of the spike protein, your cell destroys the mRNA from the vaccine. It's important to know that the mRNA from the vaccine never enters the cell's nucleus or changes its DNA in any way. Next, your cell presents the piece of the spike protein on its surface. This allows your immune cells to detect the protein and recognize that it doesn't belong there. As a result, your immune cells begin making antibodies as part of an immune response to the virus. In the future, if you catch the virus, the antibodies recognize and attach to the spike protein pieces on infected cells and the spike proteins on the virus. This marks them for immediate destruction by other immune cells. Like all vaccines, the benefit of these mRNA vaccines is that they give vaccinated people protection from the virus without having to get sick with COVID-19. Most mRNA vaccines for COVID-19 require you to get a second shot within a few weeks. Sometime after getting the vaccine, you may have symptoms such as a fever. This is normal. It means the vaccine is working to make you immune to the virus. Vaccines protect you, your family, and your community from diseases that can be dangerous or even deadly.
It seemed nothing short of miraculous. mRNA vaccines approved for human use for the first time brought the end of the COVID-19 pandemic into sight less than a year after it had been declared. Shattering records for development, the Moderna and Pfizer-BioNTech vaccines were conceived, created, tested, and approved for emergency use in months, not years. But that feat was made possible by decades of foundational work by hundreds of scientists, whose combined efforts created platforms upon which the mRNA vaccines were swiftly built.
The coronavirus vaccines and how quickly they became available was just really an incredible example how when scientists, policymakers, manufacturers, industry people come together and the government supports, puts in the resources to be able to do this, how quickly you can go from zero to vaccinated. But what people I don't think appreciate is that mRNA technology has been in the laboratories and clinical trials for a decade, even longer, and work on coronavirus to understand what would be the protein that you would target. That's been happening since SARS first emerged in 2003.
Well, I think it's a really interesting story when you go back on the mRNA development over decades, because a couple of the pioneers, Catalin Carrico and Drew Weissman, they had really struggled. They couldn't get grants. They had a lot of naysayers about their efforts. But really it was that basic science investment, which wasn't really mission driven, that was the foundation for this rapid success.
All these things that people have been working on for 30 years have now exploded into the success around this vaccine and the ability to do it quickly. And all of these things wouldn't have been happening if it weren't for all the work that everybody has been doing for the past 20 to 30 years.
I mean, think about it. It's the first truly new vaccination technology in decades. If you had told me, as you said a year ago, that we're going to use mRNA as a vaccine platform, I would have said that's not going to work because the mRNA is just going to get degraded. But the companies came up with a trick of encapsulating it in these nanoparticles, and those nanoparticles are designed in a way that cells take them up. So that's the trick.
Pandemic urgency gave mRNA vaccines the chance to prove themselves. Having passed this critical test, this adaptable platform could be exploited to protect against other disease threats.
Today, we need methodologies that we can rapidly adapt so that when a new pathogen arises, we can make a vaccine against it. And those types of methodologies are what we refer to as platform technologies.
Well, it's a curse and an opportunity at the same time to move forward technologies that probably we were seeing with a little bit of hesitation, which is the messenger RNA vaccine technology. Something that has been, research has been done and proven that there was a profile of safety, but the pandemic certainly accelerated our interest into responding quickly to it and developing this technology for the SARS-CoV-2 pandemic. we can apply the same technology for viruses that are in chickens or for viruses that are in humans. Okay?
I think it's opening a lot of exciting possibilities for other vaccines. To be able to use the mRNA technology, all you need to know is a sequence, and you can then create this mRNA-based vaccine to almost anything and do it rapidly.
I am fairly certain that you're going to see more widespread use of mRNA-based vaccines because they've been shown to be so effective. I suspect that you may also see some further engineering of these vaccines so that they remain immunogenic.
I do think now that we have new vaccine platform technologies, we will change the way we think about emerging infectious diseases, and I don't think that we're going to be left in such a position with future emerging infectious disease emergencies.
What we've now shown with this pandemic is that we can be very fast at developing vaccines for novel pathogens, at least this novel pathogen, which may have been a relatively easy virus against which to develop a vaccine, but nevertheless we did it in about 300 days between the sequence availability and data supporting use of the product. So the question is, what tools do we now have at our disposal that can allow us to shorten the timeline even further? And here the mRNA platforms provide a tremendous opportunity for us to identify viruses of interest ahead of time, develop candidate vaccines against those viruses, ideally broadly effective vaccines so that multiple viruses within a given family would be addressed by that vaccine, and do that across multiple families so that you have a toolkit of vaccines that are ready to be tested in an efficacy trial once a pandemic emerges, if you were so lucky as to have a candidate vaccine against the virus that emerges.
Influenza is a mutable virus that takes hundreds of thousands of lives each year and has caused several devastating pandemics over the last century, making it a prime target for the mRNA vaccine platform.
There's only a limited number of vaccine technologies that we've been using to create influenza vaccines. We have eggs, we have cell culture based, and we have recombinant proteins. Now, with some of these new vaccine platforms, the mRNA platforms and vectored platforms, that may give us more flexibility to create better influenza vaccines, and also maybe the opportunity to be able to change the vaccine mid-season if we see, a shift in what's circulating in people, because those vaccines can be manufactured very quickly and relatively inexpensively, and it's a lot easier than working with the whole virus. The system is already primed, and we've learned so much from the COVID-19 vaccines that I think we could rapidly synthesize and test an mRNA-based influenza vaccine. And if it's as immunogenic as it is with COVID-19, then again, your likelihood of it being a successful way to immunize against flu is very high.
Messenger RNA technology could allow us to make vaccines that are less affected by these things of growing the virus in tissue culture in eggs. Is that right? So you basically sequence the virus, recognize the antigen that you need, you clone it, and you don't need to express it anywhere but in the arm of the host that receives the vaccine.
There will be a time where we can probably use mRNA vaccine technologies against influenza, and you could technically print out new hemagglutinin versions every year. We want a vaccine that we don't have to update. We want to find a part of the flu vaccine that doesn't change, so you don't have to update it. We want to be able to be kind of one and done the way we are with measles or mumps or rubella, other viruses for which we have vaccines.
When I dream about these vaccines late at night, I'd want a vaccine that ideally you'd only give us one shot, but I don't think that's realistic. I think it's probably more realistic that we would give one shot every 3 to 5 years, and I'd be quite happy with that.
The mRNA does appear to have the ultimate flexibility. You go from sequence to template in a matter of hours, really. So, I think this is the front runner. It's got proof now. It's got incredible momentum and experience. So it's really ideal for the universal vaccines of the future.
I actually think that the advent of vaccine development after COVID will allow these new technologies, or newer technologies, because some of them have been around for quite a number of years, help push the development of making a universal influenza vaccine further than we've been able to achieve in the last 10, maybe 15 years.
Researchers had long recognized the strategic potential of mRNA-based influenza vaccines, but lacked the necessary resources to carry the product across the R&D finish line. Now, with greater financial support and the vast evidence base created during COVID-19, those plans are moving forward.
So in the case of mRNA vaccines, we have a platform that until COVID had shown a lot of promise. There had been programs taken all the way up through phase 2 development, but there had never been a phase 3 efficacy trial conducted with these vaccines because there either wasn't enough capital available to make that investment, risk tolerance to do that, or disease available, circulating disease available to actually conduct an efficacy trial, or some combination thereof. I think it is, it's a platform that we absolutely should pursue for flu. Look, I think that mRNA has become the new default technology to apply to every pathogen. And I think you hammer everything with mRNA approaches to see if you can make it work. And only when you show that you can't, despite a lot of, optimization of all these parameters, then do you look for other approaches. But I think it's table stakes is mRNA for everything, including flu.
Simpler and far quicker to make than their traditional counterparts, mRNA vaccines could potentially be produced in many countries where vaccines are not currently manufactured.
I think that we're in a unique moment in time with the demonstration of the mRNAs to give countries a way to leapfrog existing vaccine manufacturing, as well as a gateway to more traditional manufacturing.
I think we need to make sure that we make the manufacturing process as easy as we can, so that it can be transferred to low to middle income countries. And I think that is probably something that we should do with all vaccine technologies, that all low to middle income countries, and in fact all countries, should have the ability to make vaccines for themselves and to supply worldwide.
mRNA vaccines could be key to quashing future viral outbreaks before they have the chance to become pandemics. Decentralized vaccine manufacturing would make this happen even faster, a goal that agile mRNA vaccine technology brings within reach.
I think coming out of this pandemic, it's a huge lesson learned for many countries and regions. They're going to have a lot of demand for this kind of capacity building. On a positive, very positive note, we do have some new technologies that can help here, and I think the mRNAs vaccines are the best example. These are chemistry-based approaches. I believe that they can be more amenable to introduction to countries as platforms that they can build an ecosystem to support. And if they can get off the ground with mRNA or chemistry-based approaches to vaccine manufacturing, I think that can then serve as a gateway to the more complex traditional vaccine manufacturing.
mRNA exemplifies the emerging technology that can not only lead us out of this pandemic, but could stop the next one, if we invest now in applying, refining, and distributing it worldwide.
I think of R&D as a machine where you put in about $1, so you put in $1 and it spits back at least $5. So the greatest investment machine in the universe. You put in $1, it's going to give you at least $5 back on average. It's incredible. The reason innovation produces such a high return is because of its spillovers. It's because the benefits it creates bring benefit to so many different people in so many different pockets, if you will. Okay, so for example, let's take something very concrete in our minds, a COVID vaccine. But the value of the lives saved is, the value of that is in excess of what people are paying for that vaccine. The value of starting up the economy again is so greatly in excess of the cost of that vaccine. We're talking trillions of dollars.
I think we have in our hands today the power to protect all future generations from pandemics. I mean, that's incredible. I mean, this is the biggest accomplishment of the century. And it happened, right below our noses. I mean, we were sitting around dealing with this pandemic, and boom, what unfolded in front of us was this roadmap to prevent the next pandemic, and nobody can ever take that away from us.